And so I'd first like to introduce Dr. Suneel Apte from the Cleveland Clinic, who's going to be talking about forward and reverse degradomics of cardiovascular extracellular matrix.
Thank you, Kathy. I began my career as an orthopedic surgeon, and then I became an extracellular matrix molecular biologist just across the street here. And now we study cardiovascular disease thanks to the investment made by the American Heart Association jointly with the Allen Frontiers group. And in essence, these tissues, like every other tissue in the body, share a phenomenon of cells embedded in extracellular matrix, as you can see in this mosaic that I've used to represent it here. And there are actually remarkable similarities between the musculoskeletal extracellular matrix and cardiovascular extracellular matrix because, in essence, both are mechanical tissues. So in the mosaic, the tiles represent the cells, and what's in between them is matrix, and it's static and has been this way for a couple of thousand years in ancient Greece. But in reality, in our bodies, the matrix is constantly in flux, and it's an exceptionally dynamic entity, but what happens in the matrix doesn't stay only in the matrix. It affects every aspect of cell behavior of those cells that are embedded within it. So I'd just like to specify very briefly that whereas we often think of a genome as encoding a proteome, it actually encodes numerous proteomes. And that's because of the phenomenon of post-translational modification, which can affect every gene product that we know of. And the one that I'm going to focus on today is proteolysis, which is unique in the sense that it's irreversible. You cannot take two cleaved parts of a molecule and put them back together. And this cleavage process both has regulatory roles in terms of activating molecules, but also in terms of destroying functional entities. So the branch of proteomics that deals with extracellular matrix and cellular proteolysis is called degradomics. Really the biology of a protease is the biology of its substrates. So if you want to understand what proteases do in tissues, you have to identify their substrates, and this is what degradomics strategies entail, and I'll explain those to you in a moment. But the effect of substrate cleavage is twofold. You can either get a positive effect, which is you can create new proteolytically derived fragments that have new functions, or you can destroy the function of a molecule and inactivate it. We've termed degradomics approaches with two new terms. We call reverse degradomics the approach whereby we test the effect of genetic inactivation of a protease one at a time. And then forward degradomics is an approach in which we can take a diseased tissue and look at the entire proteolytic landscape in a systems approach. And what I'd like to show you here are examples of both of these approaches applied to cardiovascular disease and cardiovascular developmental defects. So proteomics suffers from the needle in the haystack problem. You cannot find a needle in this haystack. But you can if you make the haystack very small or you make the needle much larger. So very germane to effective application of proteomics is a good sample workflow that enhances for the things that you'd like to see. And so the approach that was pioneered by Chris Overall and Ulrich auf M. Keller and colleagues less than a decade ago is called TAILS. And it relies on two very important but simple steps in a proteomics workflow. You, of course, isolate proteins as you normally would, but you label them at the protein level with reagents that bind to the amine terminus or to the epsilon amine of lysines, and then you trypsinize them to develop the peptides that you typically inject into a mass spectrometer. These cleaved peptides are then put through a workflow that binds to all of the unblocked and unlabeled peptides and eliminates 90% of those typically. And then you take the remnants, which represent all of the N-termini that were either native N-termini of proteins or new N-termini created by proteolysis, and you inject them into a sensitive high-resolution mass spectrometer. The one we use is a Orbitrap Fusion Lumos. So we've applied this approach in our forward degradomics strategy to a variety of cardiovascular diseases that are listed here. And I'm just going to show you a couple of examples. Most of these are in humans because we have the good fortune at the Cleveland Clinic to have collaborations with cardiothoracic surgeons and an exceptional caseload of cardiovascular disease. But in addition, we are also undertaking a fundamental approach, which is determining the entire landscape of proteolytic turnover during mouse embryonic heart development. So I'm going to talk to you about a couple of these things. And I actually need to switch out from projection mode because I just realized that when I was reviewing the slides, that one of the slides I'm going to use is, can I do this, Kathy? No. Okay, so I'm just going to stick on with this. Yep. So this is an example of some of the methods we use and how they work. What you see up here is a Venn diagram that illustrates the number of N-terminally labeled peptides isolated in a routine mass spec scan. But when you enrich, you pull out a substantially larger number of N-termini. And then depending on the modification that you find at the N-terminus, you can assign those to either natural N-termini or newly derived proteolytic N-termini. And so, for example, in this analysis that we did with abdominal aortic aneurysms, we could find a large number of proteolytically derived peptides that are either more abundant in AAAs or less abundant. This is a very common disease in the elderly population and can result in catastrophic rupture of the aorta. So in a sense, it is a disease that's characterized by extensive remodeling of cells and extracellular matrix. And we can take these differences that we find between the diseased and the control aorta and do a gene ontology type of analysis and determine which specific pathways are perturbed, for example, in the aortic aneurysm versus normal tissue. And so what you see here, for example, are pathways that are more active in AAAs, acute phase response signaling, LXR, RXR activation, whereas the control aortas tend to have higher incidence of other pathways that indicate a much more healthy interaction between the cells and the extracellular matrix. So we've done this kind of analysis now also for thoracic aortic aneurysms, which actually represent a completely different disease cohort, as well as control normal thoracic aorta. So we can now do four-way comparisons between the abdominal and thoracic aorta and the diseased aneurysm counterparts to understand a bit more about this disease. So the reverse degradomics part relates to a very important family of proteases called the ADAMTS proteases. This family is extraordinarily important in a variety of human diseases, including cardiovascular diseases. And nearly every one of these proteases has a developmental defect. And I'm going to speak particularly about two groups of proteases, ADAMTS 9 and 20, which form a homologous pair, and ADAMTS 6 and 10, which form a second homologous pair. Both are important in cardiovascular development. So a few years ago, we found that the mice with combined deficiency of ADAMTS 9 and 20 had a very complex morphogenetic defect with defects of the neural tube, limb development, axial asymmetry, and cardiac involvement. And it turned out that these secreted matrix-degrading proteases actually had a role in development of a primary cilium. So what you see here are scanning electron micrographs of a normal neural tube that's been cut open with a cilium on more or less every cell on the surface neuroepithelium, whereas in the mutant, either cilia are absent or are very short. And so to cut a long story short, because this was published recently, we know that these proteases, despite being secreted, are internalized into the cell and targeted to vesicles that live at the base of the primary cilium. So for those of you not familiar with the cilium, it's a unique and non-motile structure that protrudes like an antenna from the surface of the cell. It is made up of microtubule and a large number of very specialized and associated proteins. And its growth requires the addition of membrane from vesicles that collect at the base of the cilium. And one of those population of vesicles is enriched in ADAMTS9 and ADAMTS20. So this cilium is really important for sonic hedgehog signaling in the embryonic period, and these mice mutants show defects in sonic hedgehog signaling. So obviously, at a protein mechanism level, we'd like to know what the substrate of ADAMTS9 and 20 is in this context. And so we generated, for example, RP1 cell lines as well as another cell line that was defective in ADAMTS9 and did not express ADAMTS20 at all. So we had a mouse, a human cell line that was defective in both ADAMTS9 and 20. And we subjected these to analysis to verify that they did not produce a cilium. So you can see that these cells in the wild type cells, RPE1 cells, make a good primary cilium, and the mutants don't. And then we applied this degradomics strategy to understand what was different at the substrate level between these two populations of cells. And so this is an example of the kind of peptide you get. It's labeled at the N-terminus with an iTrack reagent. You can do very precise quantitative analysis to find out what the relative abundance in the wild type and the mutant is. And in this case, this particular peptide was completely absent in the mutant cells subjected to the analysis. Where does it come from? It comes from a very important cell surface membrane integrated protease called MT1-MMP, which is of exceptionally high importance in general in biology. And this was a great surprise to us because this enzyme is primarily involved in turnover of extracellular matrix. So whereas ADAMTS9 and 20 do cleave extracellular matrix and they are targeted to the base of the cilium, it's somewhat of a paradox that the one major substrate we seem to have identified is primarily involved in remodeling of the extracellular matrix. And that raises a number of questions about how important the extracellular matrix may be in regulating ciliogenesis. So to validate this, we looked at the proteolytic shedding of this protease from wild-type cells, where you see the expected fragment here, whereas the mutant cells make undetectable or very low amounts of the shed form of this protease. In addition, if you overexpress ADAMTS9 with MT1-MMP, you get shedding not just of that 22 kilodalton band I showed you earlier, but also of the entire ectodomain. So ADAMTS9 is secreted, binds to the cell surface, and has a major role in shedding the entire MT1-MMP ectodomain. Remember that the mutants that I showed you have a defect in both ADAMTS9 and ADAMTS20. So one requirement for this being an effective substrate that explains the defect in ciliogenesis is that it should also be cleaved by ADAMTS20, and that's exactly what we see. So this sums up, in a sense, some very unexpected findings. We get cleavage of this protease that essentially destroys its ability to cleave at the extracellular surface. And genetically, inactivation of this protease, which coincidentally I performed 20 years ago when I was a postdoc here across the street, is really not a good model for understanding the role of this protease in ciliogenesis, because what we see now is an excess of MT1-MMP in the cell. The second vignette I'd like to conclude with is of ADAMTS6 mutant mice. These mice have a variety of defects, but the gene is very highly expressed in the heart, as you can see on the right by the red in situ hybridization signal. And in fact, these null mutants, which were generated by Cecilia Lowe's lab, have profound cardiovascular defects, such as a double outlet right ventricle and overriding aorta, ventricular septal defect, and so on. So we applied the degradomics strategy to this as well, and identified fibrillin-2, which is a major extracellular matrix microfibril forming component, as a potential substrate. The cleavage site occurs between domains, which is An important realization, because cleavage in a disulfide bonded domain shouldn't partition a protein as effectively as cleavage that is interdomain. And in fact, this cleavage is very important because if you culture fibroblasts and allow them to make fibrillin 2 microfibrils, which you see here, and you do that then in the presence of a catalytically active ADAMTS6, you don't get any microfibrils formed. So clearly, this enzyme is able to degrade microfibrils, and we've been able to show that it also degrades monomeric fibronectin, fibrillin 2, by biochemical assays. So what is the in vivo evidence that fibrillin 2 is a major substrate for ADAMTS6? You see here fibrillin 2 staining in wild type and ADAMTS6 and ADAMTS10 mutants. Just as ADAMTS9 and 20 are a homologous pair, ADAMTS6 and ADAMTS10 are also highly homologous, and both of them now are known to cleave fibrillin 2. What you see here is accumulation of fibrillin 2 in ADAMTS10 mutant outflow tract. These are the aortic valves. This is the aorta. In the ADAMTS6 mutant, there is a great deal more fibrillin 2. And in the combined mutants of these mice, there is considerably more fibrillin 2. And the amazing thing is that you can potentially reverse, and we do reverse the majority of the musculoskeletal defects and some of the cardiovascular defects simply by taking the ADAMTS6 mutant mice and deleting one copy of fibrillin 2. So it does strongly suggest that an excess of uncleaved fibrillin 2 is responsible for the morphogenetic defect in these mice. So I'd like to sum up everything that I've told you by pointing out that these kind of approaches are invaluable for understanding the pathways and processes that take place both in diseased tissues and in genetic defects at the level of tissue extracellular matrix turnover. And I think they inform us about potentially new pathways that can be targeted for therapy, but they also provide extremely valuable biomarkers for disease. For example, in the case of our applications to aortic aneurysms, one of the major challenges is understanding how rapidly the aneurysm grows and who should have aortic replacement surgery and when. The same holds true for cardiac failure, where we find numerous cleaved peptides that are present in pressure overloaded hearts. In infective endocarditis, where we see products of both the microbial organisms that infect the heart and the products that form vegetations that could serve as biomarkers. So I'd like to conclude by thanking my colleagues at the Cleveland Clinic, both in my lab, you see on the left, and an exceptional cohort of clinicians we work with who make a very important contribution to our work. And most of all, to the Paul G. Allen Frontiers Group and to the American Heart Association for investing in this work. Thank you very much.